Hi, my name is Maggie, and over the past couple of weeks, I've been working on a project where I built a kill switch using a software program called Synthetic Biology Open Language. And before I jump into that, I want to just talk about just how incredible and fascinating gene editing is, and the fact that now that we have tools such as CRISPR and prime editing, the potential for us to manipulate DNA in order to create novel solutions to current problems is incredible, as gene editing can impact so many different areas, including medicine, agriculture, and industry. And if we're talking specifically about genetically modifying microbes such as bacteria, that includes potential applications as developing new medicines and treatments using bacteria for bioremediation purposes, or even using bacteria to create new bioprocesses that replace current industrial processes that use up less energy and less resources. But it's important that while we're considering all these future applications and how we want to develop these new solutions to either improve the climate or create new medicines to cure certain diseases, it's important that we consider certain safety mechanisms and certain limitations that we currently have in order to ensure that this technology is creating the most positive impact and that it's not causing any sort of environmental harm or serious impact on human health. So one specific thing that I was researching, which is kind of the core purpose of this project, was kill switches. And kill switches are basically biological safety mechanisms that are put into bacteria, for example. And when the bacteria is exposed to certain environmental signals, it causes the bacteria to commit cell suicide. So how exactly does this work then? So for my project, I based it off this research paper, and these are kind of the results of what I built in SBOL. So these are just screenshots. So on the left, it just shows the two different components, and on the right, it kind of shows the different parts that make up these components. So it's like a second level view of it. So we have three main parts, which is a promoter, genes, and terminator. And a promoter is basically where transcription begins, and it's where something called RNA polymerase, or RNAP, binds to initiate transcription. Next we have our genes, which are what is going to be expressed, and usually when genes are expressed, they create proteins. And finally, we have a terminator, which basically tells the RNAP where to stop transcription. So this is kind of just an overview of how the entire system kind of works, where we have our survival and death states, we have our different genes, and then we also have special biomolecules, which are IPTG and ATC, which allow for us to switch between the different states. So when we're talking about TET-R and LAC-I, they are repressor proteins, and they mutually repress each other, meaning that when one is expressed, it will repress the other. So for example, if we have TET-R being expressed, it will repress LAC-I, meaning that only mCherry will end up being expressed and the toxin won't end up being expressed. And the inverse is when LAC-I is being expressed, it will repress TET-R. So in this case, now we have the toxin being produced. However, things become interesting when we add specific biomolecules. So let's say we're originally in the LAC-I state and we're producing some toxin. And because LAC-I is being expressed, it will repress TET-R. However, if we add IPTG in the system, it will repress LAC-I. And without LAC-I being able to repress TET-R, that means TET-R will now be able to be expressed and repress LAC-I, which will stop the toxin from accumulating and instead production of mCherry will begin. The same thing happens if we were originally in the TET-R state, or the survival state, where we're repressing LAC-I. However, when we add ATC to the system, it stops TET-R from repressing LAC-I, and as a result, LAC-I will end up repressing TET-R, and toxin production will begin. So if you got to the end of this video, thank you so much for spending this time with me. If you're interested and you want to learn more, I've included a Notion project page link in the description box below, so you can feel free to look through the resources that I used while I was in the process of doing this project, as well as some of the notes that I took along the way. Also, if you're interested in keeping in touch, feel free to either connect with me on LinkedIn, or you can even send an email to my personal email if you have any sort of questions, and I'll try my best to answer them, since I'm still in the process of learning as well. You can also sign up for my personal newsletter, which is just a monthly update of things that I've been working on, interesting content gems I found along the way, and so forth. So thank you so much, and I'll see you next time. Bye!